Hello, in this video I will be solving past paper questions from the chapter Enzymes. I tried my best to explain the answers. I hope you guys get an idea on how to tackle the paper by the end of the video. I have selected the questions from papers over different years, paper 4 extended, and that are relevant to the topic. I hope you forgive me for the audio. Try to use a headphone when listening to this. Oh, and also jot down notes as I move to different slides. An experiment was carried out to investigate the effect of pH on enzyme action. The graph shows the results. What are the labels for x-axis and y-axis? We should know that in x-axis we only put in independent variables that we are in control of. So right now we are investigating the effect of pH on the enzyme action. So this would be our independent variable. So I'm left with option A and option B. And then on y-axis, it could either be rate of reaction and time. Now, rate of reaction is the speed at which the reactants are converted into products. So this is basically me finding out if the enzyme is working or not working. So time is just useless putting in there, will not really get any crucial information. So the answer would be A. The reactions of chemical digestion are catalyzed by enzyme. The figure shows the stages of enzyme catalyzed reaction. So we have this as A, this space right here is B, and then this whole thing as C, and then tiny bits as B. So this would be substrate, active site, complex, and the product. Right here I've written the answers: A as substrate, B as active site, C enzyme substrate complex, D product. The apparatus shown is used for an experiment on starch digestion. Which test tube contains the most sugar after 20 minutes? So, since we're experimenting on starch digestion, I should expect an amylase enzyme in the test tube. So, this one's got an amylase along with the substrate that we're breaking down. This one's also got amylase along with the substrate. So it could either be A or D, not B and C. B and C, nothing will change in it. There is no enzyme to break it down. After so long, you'll still find the same starch molecules in them. Now, the only difference between these two beakers is that one is at water bath at 15 degrees Celsius and the other is 37 degrees Celsius. We know that at higher temperatures, this amylase, the enzymes, they work faster. So after 20 minutes, we'll find more sugar in this beaker rather than in the beaker with 15 degrees Celsius. So the answer should be D. Next question. Young mammals that are often can be bottle fed. The figure shows a newborn tiger cub sucking on a bottle. So let's start underlining. The digestive systems of young mammals are not fully developed. Okay, then there are enzymes are often added to baby food to aid chemical digestion. Fine. Then they are saying to state the substratum products for each enzyme reaction. So amylase, maltase, and protease. This shall be easy if you have studied and watched my previous videos. Substrate of amylase would be starch products, glucose or maltose. And then for the substrate for maltase would be maltose into glucose, and for protease would be protein into amino acids. Right here I've written it down. The next question says, suggest why the temperature of baby food must be controlled when the enzymes are added. The thing is, we know that enzymes, they get denatured when the temperature increases, so a set temperature is needed for the enzymes to work their best. So the answer is, we will write all the possible points. Just for 2 marks, I'll write 3 points. It maximizes the chances of you getting correct answers. So we have high temperature denaturing the enzyme, and why we should not use low temperature, because that would lead to slow reaction. And we should also say that we need to control and give them the optimum temperature that they can work best at. Next question. State one other condition that must also be controlled to optimize enzyme activity. The other factor that controls the enzyme activity would also be pH. So you can write pH. You could also write enzymes concentration, which means more enzymes, to break down all substrates real fast. Next question. Which graph shows the effect of temperature on the activity of an enzyme? Let's look at C and D. This makes no sense to be an enzyme graph for the activity to decrease and then come back alive. It's like denaturing and then coming back alive. So nope, nope. Now in A, it shows that as the temperature increases, the activity increases, and then there's sudden steep line. That's not how it works. You have to know that it curves out like this, then slowly it gets denatured and done. So answer is B. The diagram represents a chemical reaction. Which diagram represents the enzyme for this reaction? So we have two products that are supposed to fit into one active site and form this product. So I will see whichever active site is matching the product for the reactant. So this will be the second reactant, and this should fit the first reactant, and then the enzyme's work is to form the bonds between these guys and then make this as the final product. So the answer is D. The table shows the conditions in 4 test tubes containing equal amounts of starch and salivary amylase. In which test tube is the starch broken down fastest? So amylase, they work best in alkaline conditions. Alkaline conditions would be 7, 7, not 2 and 2. pH 2 is too acidic, it will get denatured, so we are left with the option C and D. Temperature wise, we know we should go with something that's warmer, so it can work better. Answer would be D. Which group of compounds ensures that metabolic reactions take place effectively? For a metabolic reaction to take place, carbohydrates has nothing to do with, they are just nutrients. We'll come to that. Fatty acids are also, like I said, byproducts of the nutrients being broken down. Hormones, they do nothing in metabolic reactions. They are just some chemicals that signals an organ to do something. Answer would be enzymes, obviously. They are the ones involved in breaking down and doing everything to do with metabolism. So answer is B. The graph shows the effect of pH on a particular enzyme controlled reaction. When is the enzyme not active? So we can look on the basis of the rate of reaction. So at pH 1, the rate is 0. That means the enzyme is not working at all, or the speed is too slow. No reactants are being converted into products, even right here at pH 13. So the answer is A. An experiment was carried out to investigate the effect of pH on enzyme action. The graph shows the results. What are the labels for x-axis and y-axis? Again, effect of pH on enzyme action. The effect of pH would be on the bottom. Enzyme action, in the sense activity or the rate of reaction, would be here. So this is right? Yeah, A. What is the characteristic of all catalysts? Are they broken down in the reaction? No. Are they made of protein? Yep. Not really. Not all the catalysts. We have catalysts made of metals, like not made of metals, that are metals, like iron. So not all of them are proteins. Just the ones in our body are proteins. So nope. They are not changed by the reaction. Yes, we know that. They do not change the rate of reaction. What's the purpose of catalyst? To change the rate of reaction. So this answer is C. In Sichuan in China, I guess that's how it's pronounced, a sauce is made from broad bean seeds that have germinated and then have been left to ferment. So we have a germinating broad bean seed here. We're not going to label this. This is not part of the enzyme chapter. Okay, this is the question for that. The action of enzymes is often explained in terms of the lock and key model as shown. Let me underline that. Okay, fine. So it's use the information. See, whenever they use this word, use the information, better use it if you want to get the marks. explain in the sense, explain all the details on how it works. Do not just describe in short or brief. Explain how enzymes work to break down starch. So we're going to use starch as an example in the answer. So we're going to say how the substrate would fit into the enzyme when its active site right like this. Then we would say that the shape of the substrate is complementary to the shape of the active site, and that is why they are being fitted. And then we will say that the substrate is acting as the key, enzyme is acting as the lock. That's why they're saying use the terms lock and key. So this part of the point is important to write down in the answer. Then we should say that the substrate is being broken down. Which is the substrate? Starch. So I kept them in the bracket. It's broken down to products, that is maltose or glucose. And then we will say crucial things about enzymes, like they are not changed at the end of the reaction, and they can work again and again on another substrate. So that is for 4 marks. I've written 5 points, so I go ahead and write 5 or 6 points. Just make sure your answers are short and short, but that makes sense. Do not waste time writing lengthy answers. Enzymes in bean seeds are activated during germination. Fine. Some of these enzymes break down proteins stored in seeds. Large number of bean seeds were sown and germinated. Researchers took samples of germinating seeds over a period of 15 days. The seeds were chopped into small pieces, crushed with water to make an extra. Equal quantities of extracts were placed into protein solutions. pH 5, pH 8, 2 different pHs. The activity of enzyme in each extract is determined by recording how quickly the protein is broken down. So how quickly, in the sense the time it takes, how long it takes, and then their activity up here. So this line here is for pH 5, shows their activity increasing, decreasing, and then the steep increase and then a steep decrease. And then we can see on the dashed line for pH 8, there is some kind of a weird increase and then constant and increase and kind of constant, then increase, and goes on. So there's no decrease after that. So that's the two differences between pH 8 and pH 5. The question says, describe the activity of the enzymes in the extract at pH 5 over 15 days. So they just want pH 5 answers over 15 days. Do not talk about pH 8, and they're just saying to describe the activity. Describe what you see on the graph. Do not bother explaining how they work. Just say what's happening, what do you see on the graph. So the answer is this. We will talk about how up until day 5, this is day 5 right here in the between, this activity right here is too little, so very little activity until day 5, and then we will talk about this peak. Always use these answers in whichever graph you have. Talk about what peaks you see, what levels you see, and all. So this peak is right at day 11, somewhere in the middle here, and then after that sharp decrease till day 15. The researchers concluded that the beans contain 2 different enzymes that break down proteins. State the evidence. State from the graph for this conclusion. Now, how do we know that there are 2 different enzymes? So first we look at this, that protein molecules are same in there, but we can see that the enzyme activity is shown by 2 different graphs here, 2 different lines, and both of them are working completely differently at 2 different pH. So like for example in pH 8, we can see the activity being really nice, really fast, and then pH 5, the activity started at day 5 instead of day 1 or 2. So that should be a big revelation. There are 2 enzymes in there. So this is how you write the answer. You will say that there is enzyme activity in both the pH, and then we know that enzymes are specific to 1 pH. So one enzyme works for 1 pH. So the fact that there are 2 different pH, activity on 2 different pH, so 2 different enzymes, talk about that. Graph shows different lines at different pH indicating 2 different activity, and of course 2 peaks that we are seeing we have seen are shown at different days. This is due to the difference in the pH. Hence, it's clear that there are two different enzymes present. Written 4 points. You should also write 4 or 5. Define the term enzyme. Well, enzymes are catalysts and they are present in living organisms, so we'll talk about them being biological catalysts and that they are protein in nature. 2 points for 2 marks. Enzymes are used in biological washing powders. Alright, describe, not explain. Describe how the presence of these guys may increase the efficiency of washing powder in removing stains from clothes. So, the most popular stains would be oil stains, those greasy stuff that would contain fats. We need to break down fats. We know there are enzymes that work on fats and they are lipases. Then the next kind of stains would be blood. Doctors have blood spills on their coats too, so you need proteins to work on them because blood is basically protein, made of a protein molecule. So we will write the answer as stains being proteins of fats. So we need a lipase enzyme in the detergent that will break down those fat stains or the lipid stains to the respective products, that is fatty acid and glycerol. And then the protease in the detergent will break down protein to amino acids. Say all the basic descriptions that you can think of. And then this final answer is also something you should be writing about, is that the products that are formed, they are all soluble. They can be mixed in water, and that's what's easy to wash them away. Explain why the temperature of the wash needs to be carefully controlled. Temperature is important for the enzyme. High temperature will kill them. Do not use the word kill. Denatures the word. Low temperature will slow them down. So write it. High temperature denatures enzyme. They won't function well. Low temperatures enzymes will work too slow. Increasing the temperature above the low value. Now, that's how whoever they are making the enzymes in the factories work out their optimum temperature. They start increasing from low value to higher value and see at which particular temperature do they have the maximum activity at. So increase in temperature will increase the kinetic energy of the enzyme. They move fast. Chances of them colliding with the substrates and breaking them down is high, and that's why they work faster. Optimum temperature needs to be given, and if the optimum temperature is constant, they'll work best constantly. Suggest suitable temperature for a wash using biological washing powder. Explain your answers. So, temperature being these catalysts, being biological catalysts, work in our body means you should give a body temperature. That would be 37 degrees Celsius or 35, somewhere around this. Just don't give 40. The explanation is that this is the optimum temperature for enzyme activity. Seed germination. Okay, this question is a completely different question. It does not belong to the previous one. Seed germination occurs when conditions are suitable. Explain the role of enzymes. What are their purposes in being inside the seeds? So seed germination. So the answer would be that when the seeds are growing, they need some kind of energy provided to them. So you need an enzyme to break down these big molecules and provide the energy for it. So food stores in the seed are broken down by the enzymes. And the seeds contain starch, so amylase would break down starch to maltose or glucose, and glucose is used by the seed for respiration or as an energy for growth. This is another question too. The enzyme catalase is found in lettuce leaves. Student investigated the activity of this enzyme by grinding some lettuce leaves and adding them to a solution of hydrogen peroxide. Volume of oxygen produced was measured until the reaction stopped. Fine. So we have the time here. volume of oxygen collected. So catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. So if the volume of oxygen collected is getting higher, it means its activity is high. So as time proceeds, you can see the activity getting high up until somewhere here it starts curving down, and then it levels off, it plateaus. Describe the results. Describe, not explain. The results shown in the figure, you will gain credit if you use the data in your answer. In the sense, use the figures, use the numbers. Use them if they say you to use it. So volume of oxygen increases with time, that's what we see, and then it levels off right here. Use that word levels off in your description, this kind of questions, wherever it is describe the graph. Peak, increase, decrease, levels off, all these are important words to be used. And at start it increases rapidly and then slows down as shown by the curve, and here is the figures being used. So it levels off at somewhere here, 6, and use this unit please, 6 centimeter cube at about right here 90 seconds. Explain the action of enzymes during a reaction. Easy peasy. Explain what they do. So substrate fits into active site. The shape of substrate and shape of active site is complementary to each other. Use this answer in your exam sheet. Substrate gets broken down to products. If it's relevant to a question, write down what these products are and what the substrate is, and then the products leave the enzyme as it detaches. Try to avoid writing fully descriptive answers like the substrate fits into the active site of the enzyme. Just get to the point. Make it short, but make it sensible. You don't need to waste time. You need time for the other questions. Mycoprotein is a form of single-cell protein. It is produced by fungus in a fermenter. As the fungus grows in the fermenter, it produces large quantities of hyphae which are extracted and processed as shown. So we can see that there's cornstarch being broken down enzymes, then forming glucose, and this glucose is put into the fermenter so that the fungus can grow, and then all the processes of collection of good stuff from this fungus. It's in the chapter biotechnology, I believe. So the question is, name an enzyme used to digest cornstarch. Easy peasy. Amylase. Next question, explain why it is necessary to digest the cornstarch. As we can see from this flow diagram, enzyme is used to break down starch to glucose. So we know that starch is a large molecule, it's not soluble, and this guy, fungus does not secrete amylase for getting their glucose. So this is the reason why the fermenter workers, people who's working with the fermenters, they know this fungi cannot obtain their food on their own, so we need to help them and speed up the process. So we put the enzyme and break them and give the glucose for them to grow and give us the good stuff. So glucose is needed by the fungus as nutrient for respiration and their growth. 2 marks. If you benefited something from this video, I hope you give me a thumbs up and subscribe to How Coco, as I will post biology videos every week.